Welcome everybody to RahbarTalkshow. Here we want to discuss different aspects of biological aspects related to human medicine and regenerative medicine. Today, we want to discuss about the different aspects of extracellular vesicles, such as exosome in human medicine. How we can just use these particles as diagnostics particles for detection and even therapy of different pathological conditions. So please join us and see what happened. As you know, according to the recent finding, different kinds of the extracellular vesicles have been detected in biological systems. So let's see the types of the extracellular vesicles that we can use for detection and even the therapy of the different pathological conditions and diseases. Okay, Mr. Salimi, can you enumerate the types of the extracellular vesicle in biological systems?
Yes. The most widely recognized types of extracellular vesicle, or EV, include first exosomes. The size is 30 to 150 nanometers. Exosomes are formed through the inward budding of early endosome, which results in the formation of multivesicular bodies. When these multivesicular bodies fuse with the plasma membrane, the intraluminal vesicles are released as exosomes. Second, microvesicles or ectosomes. The size is 100 to 1000 nanometers. Microvesicles are directly shed from the plasma membrane through the process of membrane bleeding. This process typically regulated by calcium influx and cytoskeletal rearrangement. Third, apoptotic bodies. The size is about 1 to 5 micrometers. Apoptotic bodies are released during the process of apoptosis, when the cell undergoes programmed cell death, and this results from the cell fragmentation and are engulfed by phagocytic cells. Fourth is oncosomes. Oncosomes is a subtypes of microvesicles, vesicles released from tumor cells. They can thought to play a pivotal role in a cancer metastasis.
Great, yeah.
As totally, the formation of extracellular vesicle can complex process that involves several signaling pathway, which include endosomal pathway and plasma membrane.
Can we focus on the exosome? You mentioned that the endosomal systems is a main player in the synthesis of interluminal vesicles. There are inside the endosome, early endosome, late endosome, and multivesicular bodies, as I know. And so, how the cell can just decide to release the ILVS or exosome to out of cell or orient them into the lysosome? Do you have any recent data about it or not?
I don't have a lot of information about your question. If it is possible, I a little think about this.
Yeah, but actually, this is a gap of science. Yes. The future study should just address how the cells, the host cells, parent cells decide to accelerate the ILVS intraluminal vesicle out of the cells or orient them to the lysosomal degradation. We don't know really how it can be possible, and so it is ambiguous. And so many the effectors and unknown effectors should be just involved in this process. Okay, thank you. Currently, there are several modalities to just isolate the exosome from biological systems. But this is a really challenging issue. So let's, the different methods we can just use for isolation of extracellular vesicles from human or animal sample, or even in laboratory setting. Farzin?
Yeah, sure. Actually, there is no any gold standard for methods that is which one is the best or which one is the worst. And actually, there isn't any gold standard, but there are some known techniques that we can easily use them to extract exosomes. One of the most important methods is ultracentrifugation, which is widely used, and it's like a gold standard. It's easy to apply, but it has some problems in it. Lack of purity is one of the disadvantages of this method. Also, another method is ultrafiltration. It has another kind of use. It's used on some cell-based medias. It somehow has a difficult technology. Also, there are some precipitation methods. Also, there's size exclusive chromatography methods, microfluidics technology, and also immunoaffinity based methods, which use some kinds of extra tools like magnetic beads. It's a little bit complicated. It's expensive. It's not that much easy to use and apply. Also, the technology is not completely standard widely, so it needs a really high amount of money, also time and precipitation.
Okay, great. Let's focus on the ultracentrifugation, as you mentioned, as a first method that and is a golden standard for isolation of the extra vesicle from the biological system. Can just explain how this system works?
Yeah, it is one of the most known and actually one of the basic forms of isolation of exosomes. And actually, it's widely used. It's easy to use, and they use fluid, the fluid of cells in it, and but it has some disadvantage because we can't diagnose the exact type.
Sorry, I'm going to interrupt you. You mean that some of the gravity forces that, I don't know, used for the isolation of the extracellular vesicle in ultracentrifugation, I mean.
Yeah, of course, and also the speed of the rotor and the the density of the fluid is important, but unfortunately, the results are not always that what we want because it's not pure and we can't diagnose the types of exosomes by size easily in this method.
As you're right. Yeah, you are exactly right. Different types of the particles, nanocyte particles, as well as the exosome, like apoptotic bodies. Some of the small-sized apoptotic bodies could be just collected contaminantly by the isolation of exosomes. So this is one of the most disadvantages of this method. Yeah, great. And also we have some impurity. And we could not just, absolutely. Great, thank you. Let's turn to the fact that how we can just immunophenotype or characterize our isolated exosome or EBS from different sampling system. Okay, Mrs. Haddadi.
Yes, recently several characterization and validation methods have been developed for both research and clinical purposes to analyze the exosome purity and for qualify exosomal cargo. These techniques include electron microscopy or some kind of techniques like DLS, RPS, flow cytometry, Western blot, and so on. I can explain.
Great. You mean that some of the physicochemical assays can be just exploited to just detect the intensity of the identity of our isolated particles, I mean IVS or exosomes. So they are just related to the proteomics and genomics, and even that is some other methods that are based on the physical properties like SEM and TM. And about, I don't know, proteomics assay, if you can just add notions about this in regard, all of it.
Yes, this proteomic methods, it's based on protein interaction or antigen-antibody interactions.
Yeah. One of the most convenient, conventional available method that can be used for the identification of exosome is based on the interaction of the antigens with our antibodies. So the most laboratory in all parts of the world and in different research centers use the antibodies for detection of the exosomal integral proteins. They should just mainly the tetraespanin. These proteins are added to the structure of the exosome during their synthesis inside the endosomes. So by targeting these target molecules using different assays, that's proteomics and even the genomic, we can just identify and immunophenotype these exosomes and they convert their lineages. One of the most advantage of the exosome is that they can just use different entering signaling pathway. They can just use different routes for internalization into the target cells. Here we want to just ask Mrs. Narimani about how these particles can just enter the target cells, by which mechanism.
The underlying mechanism is various and we can categorize them in several key pathways like endocytosis, including clathrin or caveola-mediated endocytosis, where the plasma membrane invaginates the exosomes to internalize them. In macropinocytosis, as another mechanisms, the plasma membrane extend outward to engulf the extracellular fluid and its content, including the exosomes.
Great.
As well, the exosomes can be entered the target cells by directly fusion or...
It's a physical phenomenon.
Yeah. Or it lipid rafts in plasma membrane.
Great. Thank you. Also, we have another mechanism which are involved actively in the internalization of the exosome into the target cell. For example, the close interaction between tetraespanin, the exosomal surface tetraespanin, with the receptor at on the surface of the target cells can just activate this endocytosis. Even that, we have lipid rafts. You must just be notified that these lipid rafts are also alternative pathway to just update the exosome from their surrounding. Even that these rafts are enriched in cholesterol, and they can just they absorb the exosomes and guide them into the cytosol. And so, because of the existence of different mechanism, internalization mechanism, the exosome can be directed to the target cell. And so we don't have any problems associated with delivery of the exosome to the internal side of the cell, plasma membrane. So, but depending on the different cell lineages, these mechanisms can be overactivated or they have weak roles to just uptake the exosome. So when we want to just use exosome for therapy of the different target cells, in that case, we should just which mechanism is predominantly activated in the target cell. So it is so important and should be just considered in this regard.
Dear Professor, I want to say that the target cell type or environmental condition and content components can affect the mechanism, which mechanism can be chosen by the target cell.
Great. Yeah, according to my best of my knowledge, some of the documents have confirmed the effect of environmental factors and parameters like pH and temperature in the activation of the certain types of the international mechanism. Thank you. Okay, let's just focus about the fact that one of the most challenging issue in the application of exosome in the clinical setting, even in clinical trials or some clinical experiments, that is the delivery. How we can just deliver the exosome to the target tissue? How about we are certain or we can be sure that the majority of the administrated exosome can reach to our target tissue? Mr. Nazif Gerdar wants to highlight and unveil this issue right now. Yeah.
Yeah, thank you for having me here. And after better understanding the biology of the exosome, challenges of delivering these exosomes and maintaining their presence in the target sites, injured sites, arises. Today, they have myriad problems associating with the delivering to target sites. One of them is the short half-life. Another one is the poor zeta potential, determining optimal dosage and targeting in difficulties, low bioavailability, and also immunogenicity and toxicity. We are facing with these kind of problems in delivering these kind of exosomes into the target sites and defected areas. Today, I would like to talk about the most common things about they have very short half-life indeed, and also poor zeta potential. And in the next slide, in the next times, we are going to mention some solutions in this regards.
Okay.
So let's get down to the business and talk about the half-short time. As you know, exosomes have very inherent half-short time, and when delivering to the target sites,
Can mention some predetermined time period for their half-time or life.
According to some studies, as per studies, it reported that exosomes derived from the human platelets reported they have short time around 5.5 hours in circulation in the blood. And also when we added and injected these kind of exosomes into our body via blood vessels, they are going to clean out by macrophages. This is another problems here that we are facing, macrophages. And also they have low zeta potential. This low zeta potential can cause aggregation of these exosomes prior to the injection and after injection, within the body. So one study shows that it links to the high-speed centrifugations.
How can it affect its potential? Do you have any data about it?
As I read in the studies, it is the buffer content, detergents, and pH and temperature that we use in this isolation method. It is going to add diverse effect on these extracellular vesicles.
How about the higher density forces that can just deteriorate the integrity of the exosome? Because it makes some rupture, and then some other molecule at their external surface can be just be degraded or wash out.
Yeah, and if we injected these kind of aggregations within the body, they are going to be toxic effects on the body. And also some studies shows that in the mice, overdosing with these kind of aggregation, it is going to be accumulated in the lungs and cause pulmonary emboli in the lungs. And
so you mentioned that it's going to reduce the retention time, as I understand. Yeah, retention time will be reduced because aggregation. Yeah, you're right. According to some of the facts, recent facts, aggregations can accelerate the phagocytosis process by resident cells, and so the exosome can be just washed out during the few hours after injection, absolutely into the solid tissues. Even that, in the case of the intravenously administered samples, because their circulation, they have a bulk distribution phenomenon. They can just reach to different sites, different tissues. The most and highly interfering tissue are hepatic tissue, splenic tissue, and even that pulmonary tissue, because they have a bulk vascular base, and it can just trap the exosome and extracellular vesicle, and they can lead to just repeated doses of the exosome, and this is not a good. This is not a good, actually. Any other comments?
In order to solve these problems, I would like to suggest one pragmatic approach. The only things that we surmount these diverse matters of the cleaning out from our body is that we need to engineer these exosomes before injection. While we can only direct inject to the, for example, for the heart we can use, or for the spleen, or for the lungs we can directly inject these kind of exosomes into the organ, into the tissue. And in this case, the immune response is going to be decreased and they're not going to wash out. But it is not the things that we are going to have here. And we can manage the surface molecules by adding or eradicating some biomolecules on the surface.
From one aspect, you are right. Sorry for interrupted you, but from different aspects, this is not actually possible. Because of what? Because we have a phenomenon, we called it protein corona. Any manipulation on the surface of exosome can just alter their molecular signature, okay, inside the body. When we introduce them to our circulation, they can just absorb, for example, unwanted molecules, proteins. So it can just reshape their how can I say, the external surface identity. And so we could not be assured that these engineered exosomes can just
efficient.
Yeah, absolutely. And so this is our challenging. It is our challenging, and any manipulation should be just in a sophisticated manner.
This is why we have low bioavailability. And another another golden standard in this regard, in order to overcome these matters, is hydrogels, that we can creatively use hydrogels in some, for example, in the bone tissue engineering. We cannot directly inject these kind of exosomes into the defected areas. We need some scaffolds. We need some hydrogels in order to put it directly into the defected areas. And after that, we are going to see what is going to happen. I think that hydrogels right now is the golden standard for...
Not actually a golden standard. We should just, get some the FDA approved for, their application in certain circumstances. But I think it's a long way to just find out which, to describe the very efficient method for delivery of exosome to the target tissue. Okay, as Mrs. Bazar, as Mr. Nazif Gerdar mentioned, we could not directly apply exosome, our isolate particles, into the target tissue. For example, bones, they have their own character. For example, compared to the cardiac tissue or muscles, for example, we could not, because in case of the, for example, fracture, we can just inject them directly to the fracture size. It is not actually impossible. Do you have any suggestion?
Yes, absolutely. As my friend mentioned it, exosomes have some disadvantage. That one of the most important them is short half-life. There's many different platforms for overcome this problems and disadvantage. One of the most important and one of the most interesting in this area is using from hydrogel. That can be natural, synthetic, or composite. Also, we can load this exosome on surface or dip on the scaffolds. As you say, we can't directly injected in some damage, such as bone fraction or the large skin damage, and we need a base for targeted delivery, a base for carrier this exosome to the damaged area, which hydrogels help us to prepare a bed for load and carrier the exosomes. Also, the different properties of hydrogel, such as mechanical, physical, chemical, or degradation of hydrogel, can affect the effectiveness of exosomes.
Exosome delivery. Yes. Yeah, absolutely. Another strategy to just use a hydrogel is that in some cases, when we want to directly inject the exosome, some of the mechanical pressure that during our, for example, injection procedure can just lead to direct injury to the exosomal structure. So using a supporting material can just, degrade, or it can just reduce any possible mechanical injury. Am I right?
Yes, also we have some hydrogel as injectable hydrogels, just filling the area. We don't have the shape, the main shape before injection. And when you have a bone fraction or injury in some tissue of body, you injected the hydrogels, and the hydrogels give the shape of injury.
They have a filling capacity. Yeah.
Yes, and then fill the area. It can so helpful for us because we can know the shape of damage and fill this area, and then by degradation and regeneration again, the exosomes release, and
Over time.
yes, over time we
Great.
have delivery.
Great.
May I add some comments?
Yeah, absolutely. Why not? Yeah.
The hydrogels that our friends mentioned, that they called it in situ gelation. It is some kind of thermosensitive or some kind of pH sensitive, stimuli environmentally sensitive hydrogels. These hydrogels can be mixed with the exosomes and cross-linkings before injecting to the areas. And after injection by radiation, pH or some temperatures, they are going to feel the shape of the area, and after that they can release, and they can feels like natural ECM, extracellular matrix of the body, and this kind of delivery is so vital.
Great. Thank you. Thank you, Mr. Nazif Kerdal. Yeah. These are, I don't know, some sophisticated synthesis particles that can be used according to our regenerative purposes. And so, yeah, you are right, exactly right. Some pH sensitive, temperature sensitive, and other factor can be just involved in the final composite, and they can just react to the surrounding stimuli, and how can I say that? Just release the exosome to the surrounding tissue. Okay. Another interesting aspects of the exosome is that they can just be used instead of the parent cell, because these particles encompass particles that they are harboring different growth factors and cytokine. They can just release the cytokines into the target cells and target lineages. So I'm going to ask Hassam to compare the advantages of the exosome compared to their, for example, stem cell. As you know, the stem cell have been extensively used for therapy of different diseases and metabolic disorders. So I think that the exosome, without any exaggeration, will be a good option to be replaced, to be used instead of the parent cell or parent stem cell. Hassam?
It's very pretty nice and very deep question, actually. But there's a bunch of data and research also I can mention in access regard with your question. But there's a huge difference between stem cell therapy, cell therapy, and exosome delivery therapy also, and exosome combined with stem cell therapy also. There are I think 4 types of therapy and drug delivery system in the therapy, for example, methods. Okay, just in focus to continue to stem cell therapy and the extracellular vesicle or exosomes advantage in the stem cell therapy. I can say that there's in general 2 types of exosomes that's secreted by normal cell or stem cell derived exosomes. These exosomes have this advantage because they can activate the paracrine signaling cellular pathway in the cells or in the communication between the 2 cells by, for example, making tunneling nanotubes or direct injection in the area physically from outside. And also not scientifically, but we can say there's a good exosomes or bad exosome in health and disease or pathogenesis conditions. Because of that, these exosomes could carry the information. Exosomes actually are information because they carry protein, lipids, and also mRNA, cRNA, and little small bunch of segmented RNAs. But these are could be inactive or active. If they are active, they can affect in the
Sorry,
host cells.
I'm going to interrupt you. So you mentioned that some certain circumstances and pathology can just force the parent cell to secrete some kinds of the exosome with overexpression or downregulation of some certain factor inside their exosomes, and so this molecular signature can just affect their effectiveness and in progression or the inhibition of the or even the outbreaks of some pathological condition to the other cells. You mentioned like that, yeah?
Yes, exactly. For example, the exosome that is secreted from the canceric environment or the cancer cells, they have the RNAs or mRNA messenger RNAs also. They can affect to host and misregulate or dysfunction the cells to make the tumoric area and damage to the cells.
Okay, so great. You are right. You are exactly right. But my focus is not about this fact as how the police that just mentioned the advantage of exosome over compared to the exosome.
I'm going to say that. For example, in the parallel with that, this is the adverse side of the exosomes, for example. But the advantage is here because I should mention the adverse to, for example, start up the advantage. The exosomes derived from mesenchymal stem cells or zygotes or another stem cells. These exosomes, very highly potently, they can affect and not adversely, but very positively, biologically positively, affect to regulate the cells and affect their mitochondrial biogenesis very firstly. Because by making the elongation and eliminate the damaged area in the mitochondrial cells, in the host cells, they can control the cells. Actually, this is a very important and a very highly effective way to biologically and very normally we can by the force of the biology we can repair the damaged cells in general. But in continue, I can mention very specific signaling pathways and very specific methods that how they can separate and how they can in the body separate in the blood circulation, direct injections, and initially secreted by the.
But actually, it is not my target. The question. This is I am going to just highlight the application of the exosome have several advantages over the stem cell application. This is the story I want to just be addressed.
I clearly I thought
we have some time limitation. So you should be just be more specific about the items.
I mentioned that about the regulation of the cells and making the healthy cells. Yes. These exosomes derived from stem cells, they are very useful to treatment or making the cells to back up their healthy function.
Actually, great. You are right. Listen, I'm going to just add some notion about your comments. Actually, the direct application of the stem cell has faced some critical problems. For example, the most of the available and escapable stem cell vials are allogenics, and we could not just directly inject them to the patients because of the risk of and possibility of some immunocell reactivity, and so. The application of the exosome is somehow an alternative to overcome the problems. So, and the exosome are some pack of the metabolic pack of their parent stem cell, and they can just exert their therapeutic effects as similar as their parent cell. So this
they can start this cytokine storm also injection of the exosome.
Yeah, these are some critical issues we should just address in the Six Nations. Yeah, great. Irrespective of different isolation methods, even we encounter with some profound problems and a collection of them from biological system. Okay, Mr. Mehmet Nejat, can you just enumerate which kind of problems are associated with their exosome isolation from biological system?
Thank you, Doctor, for mentioning this critical issue. The problem associated with isolation of exosome have always been a drawback in therapeutic and diagnostic approaches. For example, the high complexity of biological fluids and the lack of specific exosomal marker with high expression level make the isolation very difficult, make the isolation of pure fraction of exosome very challenging. Apart from exosomes, other components, biological components, proteins, lipoproteins, nucleoproteins, complexes, and other vesicles, membranous vesicles, they also can have lipids, proteins, and
Yes, you're right. You're exactly right. But there are another problems associated with exosome isolation. As you mentioned, the complexity of biofluids also can just affect how we're going to just isolate the exosome by different modalities or technical approaches. Even that, irrespective of technical approaches, we have some difficulties to be assured that we could just control the transmission of certain pathology. I mean, that some microbes, viruses, microorganisms, because they can just transfer by exosome, even horizontally and vertically. I mean, that from the cell to cell. And so we should be certain we cultured our cells in some sterile condition, a controlled sanitary system. So as well, we don't have, as you mentioned, we don't have any, just standard protocols. How we can just, which volumes are, for example, are needed for isolation of, for example, exact number of the particles, the AVS or exosome. And so these are some questions should be answered. Even any question, Farzin?
Yeah, actually, there is something important that I want to mention in isolation and purification of the exosomes, which is really challenging. And despite lots of advancements in the research of exosomes, actually there are not, some well-known and standardized protocol for all the situations. And actually there are some variability in quantitative and qualitative results. Actually, these impurities or lack of enough technology to isolate or to purify the exosomes leads to numbers of techniques, bunch of studies, lots of research. But as a result, one of the most important problems is how to apply them into clinical use. Actually, this is the main problem because there are not such a standardized method, so the results are not that pure and that what we want. Actually needs lots of work and research to make the pathway clear about how to initiate the process of isolating and how to go through the methods, the levels of and how to actually at the end purify and extract the exosome in the exact shape, exact size, having exact cargo in it, because they are like bioshuttles. So we need to study more on them because there are lots of challenges still on this path.
Thank you for your precise comments.
Ask some questions. I would like to add some comments along with the isolation, but before the isolations, we need to mass production of these exosomes in order to examine these in the in vivo conditions, even in the human bodies. For example, for in vitro studies, we encapsulated 2 million cells per milliliters of the hydrogels in order to study their effect, their proliferation, their migration, their characterizations, functionally working or not. But how can we solve these matters? This is my questions that ticking over in my mind, and I would like to ask this question.
Actually,
Mass production of these.
mass production is a challenging item. I want to just highlight this, in our current laboratory setting. We could not just produce a exosome in large scale quantities. So this is one of the most problems. Even at the most available samples are biological samples. And so we need some donors, volunteer donors, to just dedicate their samples. And it also increased the possibility of the transmission, some microbes, some viruses, some maybe other mutations, some factors, some cytokines that going to be alter the function of the target cell. I think it does necessitate further investigation.
And also, I have another question. After passaging 3 or 4 times, sometimes cells' functionality is going to be lost, or they turn to be some wild cells, and they are going to
we call it abnormal metabolic activity.
Abnormal metabolites that are going to happen. And what if happens in these situations? And if we passage it extracellular, these cells, and then drive these cells from their patterns, what will happen?
Person, do you have?
Yeah, actually, I need to add something for the former question. I think we are just seeing the whole process like designed materials and A, B, C, and the result. It's not like that from my point of view. And actually, we have lots of misknowledge in the area. Actually, mass production is not the first thing that we have to deal with.
Then how do you want to just afford the demands of the clinics? And so we have some different patients. They want to just some of the vials
in vivo studies.
Yeah.
Now, from my point of view, I think in the first stage, we should solve the problems step by step. We can't just jump from step 3 to step 10. Even we don't have enough standardized method. We can't pure them. We can't have quantitative and qualitative,
No, absolutely. It's our priority. Just produce a very high. Yeah, you're right. Exactly.
The pure exosome. We can't, extract the exact material we want. How we can discuss about the mass production or injecting this. We have to solve the first problems.
The first problem is.
Yeah, problems goes first. We don't have in the standard.
What is the problem? First problems in your viewpoints.
Yeah, we should make sure that we will initiate a method that results in the pure and exactly the exosome with the features that we want.
Yeah, absolutely. But
all the time we have tons of exosomes with different sizes, different material, different kinds, different shapes. Even we can distinguish.
But today we have not achieved this goal, unfortunately. And so different clinicians, biologists, and researchers in all parts of the world just have worked out to just resolve this issue. But
yeah, actually, there is nothing.
I would like to say that in chemical processes, in first step, that we are going to set up a process in order to purify some biological materials. For example, enzymes, whatever it is, exosomes, then we need to have this perspective in our minds for the futuristic studies, and in order to make it from laboratory to the industrial stage. So we need to focus on this in first step too. I think that this is the problems in mass production even for the studying the in vitro or even in vivos, for example, in animal studies. Can you help me? How much cells we need to in vivos? I think more than 6 million cells.
It depends on types of tissues and extension of that injury. We should just define how many cells we could just apply, or stem cells, something like that.
But Professor, hypothetically, it's okay.
Yeah.
But in reality, we are talking about what we have on the ground.
To me, yeah.
Of course, after 100 years, there will be some methods.
I don't know. To be honest, I think that we just encounter with the heterogeneous particle sizes. As Mr. Salimi mentioned at the first of our discussion session, different types of the extracellular vesicle, like exosome, microvesicles, and absolute bodies, and even large size or small size, there are at the same how can I say, niche. And so we should just separate them using some sophisticated techniques. Even that, the big problem is that we could not just, how can I say, the isolate exome from microvesicle, because they are the same, approximately in the same diameter sizes. And so this is one of the big challenges we encountered. And so it should be just to use some, I don't know, maybe there's some combination of that techniques help us to just, yield some of the high purified.
In the area, but let me clear it with a simple sample. We are having lots of cars. Mention that exosomes are cars. We are having some types of cars that we are not aware of them, but we are going to start a Formula One race.
Yeah, absolutely.
You don't know what car we have. How can we enter to the race?
I believe in fact that there is necessity is a mother of invention, and even that we should just sense the necessity and then go ahead. Even that, but we could not stop the process of progression in application of the exosome in a clinical setting. So you are right, exactly right. But we should be notified that. We should just solve them step by step.
Yeah, step by first problem goes first.
But actually, the story is not as you say in the world. Most of the clinics in all parts of the world that just use the exosome, unfortunately, without any local approval. And this is not a good.
This is the problem.
No, yeah, it's not
ethical.
This is another problem from batch to batch quality variation.
Yeah, absolutely. Because the metabolic. Yeah, absolutely. Great, absolutely.
This is the most important.
Because any metabolic alteration is going to change the cargo sorting inside the exosome, the ILS, and so the molecular signature will be altered as well. And so this is another problem. As you mentioned, the passage in the cells in laboratory setting, for the high passages, for example, from 3 to higher passages, it's going to just affect the normal cell and somatic cell. But this story is a little different in a stem cell. So the stem cell, they have some chromosomal stability. They can just resist to the insulting condition. They can just preserve their features, and they can just produce relatively stable exosome with certain profile. So for that reason, stem cells are preferred compared to the normal cells. Okay, let's jump to another questions. As you know, the exosome have been used as a bioshuttle, natural bioshuttles or shuttles to deliver our target molecules into the tissue. Different approaches and modalities have been used to just load these factors onto the surface of exosome and inside or lumina of this exosome. Rasul?
Thank you so much. I would like to appreciate to having me here. It's my pleasure. We have the passive and active methods for the loading of factors, antibody, proteins, medications for the exosomes or any others EVs. So we have here preloading and also post-loading. For the preloading, we have some methods including transgenesis, transfections, and co-culture.
Let's just stop here. You mentioned that we're going to modulate the parent cells.
Yes.
Even sophisticated to modulate the parent cells to express some types of the proteins or genes inside the exosome or surface of the exosome. Okay.
Definitely. Yeah. Yes. This is not cost effective except co-culture. That's not cost effective, but the efficacy raised up, and we have to, before designing the project, we have to concentrate the cost effective or the efficacy of the loading. It's more important for us. So another one for the post-loading methods, which is cost effective, and also we have to care the moleculars or the types of drug, proteins, properties, then we choose which one. Also, even it's the cost effective. For the post-loading, we have the incubations, we have the
direct incubation.
Direct incubations. We have electroporation, sonication. We have the another types, pH gradient. We have the another types too, such as the synthesizes. When I'm talking about synthesizes, I mean you synthesize some special kind of moleculars you load on the surface or you load on the inside as a cargo. We have to know in designing of loading what we want to do. We want to sorting the exosomes or from the other types of exosomes, or no, we want to carrying.
Yes.
Okay, and also it is very important, some moleculars proteins properties change the exosomes EVs. So
let's to be clear here. Sorry, let's to be clear here.
Okay.
From one aspect, just we want to just load the certain factors to the surface or lumina of that exosome. That's one story. The other story is that we want to modulate chemically the surface of the exosome for target delivery. I mean, to just increase the delivery efficiencies into the target sites. So these are some different aspects, and they should be just discussed separately in the different discussion session, and we're going to just hold it in the future. But I think that most of the mentioned approaches. That the post-loading approaches, they can just affect the integrity of the exosome, unfortunately, because they are based on the physical and chemical modulatory.
Yes, absolutely. Thank you so much for this question. It's very important. Using the chemical reagents, it is somehow dangerous because maybe they create some pores on the exosomes.
It can just contribute to the leakage of the intact molecules inside
absolutely. Yes, they making these pores enable us to induce our proteins or medications inside the exosomes. But the another one, they are releasing other factors that maybe they are essential for us in the changing and engineering the pathways or something like that. So in my opinion, the physical ways are more suitable. But in physical ways also, I want to mention the best and also sometimes not the best. It's the sonication, bad sonications. But we have to care about the pulse of the sonications.
Yeah, absolutely.
And
just
also
some
they make
formulas
leakage
and time.
And one thing that it's important also the efficacy. Sonications or incubation, the time, and also the types of the drug. For example, the doxorubicin. We want to loading doxorubicin in ducks, maybe ducks from company A and company 2 efficacy are different. So we have to care about it. You're welcome.
Okay, thank you. As Mr. Rasul mentioned, we have a different loading efficiency using different preloading and postloading approaches, and the suitable method should be just selected according to our regenerative purposes. Okay, guys, I appreciate your presence to have a just discussion session here. It was really valuable, and we just debate with each other, discuss very valuable items, and I hope that we could just find good solutions for in progression or application of exosome in human medicine. Thanks a lot. According to this section content, the application of the exosome and EVS have been increased day by day, and so we should just consider the fact that it can be just replaced by stem cell therapy. So different aspects in the application of the exosome should be answered. For supporting us, please subscribe, like, and share your comments below. We will respond to any questions as soon as possible. Thanks a lot, and thanks for watching our videos.